Hi, my name is Shruthi Nagulapalli, and I'm from San Jose. My mentor is Rainy Major, and I began this project because I was interested in the intersection between neuroscience and emerging genetic therapies. In this presentation, I'll be showing genetic treatments available for Huntington's disease. Huntington's disease is a progressive neurodegenerative disorder caused by mutation in the Huntington gene. This disease is caused by a repeat of three DNA nucleotides, cytosine, adenine, and guanine, also called CAG or CAG repeats. In healthy individuals, these nucleotides are repeated approximately 20 times. In affected individuals, this sequence is repeated over 40 times, and in extreme cases, up to 800 times. These repeats can create misfolded proteins, leading to the death of neurons. They can create neuronal degeneration, called cortical atrophy, leading to symptoms of cognitive decline, involuntary movements, and memory loss. Huntington's disease is often hard to recognize without genetic testing. Additionally, current treatments only address symptoms without altering the course of the disease. As such, there is an urgent need for genetic therapies to treat this disorder. There are many emerging genetic therapies, including zinc fingers, talons, and CRISPR-Cas9. This review aims to study possible genetic treatments utilizing CRISPR for various animal models. At its core, CRISPR-Cas9 is made of two key components, a guide RNA, or gRNA, and an enzyme called Cas9. The guide RNA finds the exact location in the genome and leads the Cas9 to this target location. The Cas9 then creates a precise cut in the DNA, creating a double-stranded break. Once this break happens, the cell has to repair it, and it does this using one of two options: non-homologous end joining, or NHEJ, or homology-directed repair, HDR. NHEJ is a fast but messy fix. The cell simply tries to put the DNA back together, which can often lead to mistakes, like extra nucleotides being added or deleted. These little mistakes can shift the reading frame of a gene, which is very useful in trying to silence a harmful mutation. On the other hand, HDR is much more precise. Instead of just putting the DNA back together, the cell uses a given template to repair the cut. And specifically with HDR, we can replace a mutated gene with a healthy version. But a key limitation is that HDR happens less often and can be less accurate, making it harder to rely on. This review examines three different genetic treatment models. The first is a mouse model examining NHEJ delivery methods. This study tested gene silencing of R62 mice with over 115 CAG repeats. Researchers delivered CRISPR-Cas9 through a viral vector and attached a fluorescent protein to track levels of mutant HTT in the genome. The results revealed 40% fewer mutated protein formations and as much as 12 times as many insertions and deletions, a sign of NHEJ success. Researchers also conducted rod and rotarod bounce beam tests, finding improvements in mice as well as lifespan increases. The second is a pig model examining pigs with over 150 CAG repeats. Researchers employed HDR to replace the mutated gene sequence with a normal 20 CAG gene sequence, delivered through a striatum injection and intravenously. A fluorescent protein was delivered, finding a 50% reduction in mutated protein levels. Researchers also conducted motor tests where pigs walked along a sandy track, finding that pigs that were treated had more stable walking patterns. However, approximately 60 to 69% of edits came from insertions and deletions, not through a replaced gene sequence, which could show that results were brought about by accidental mutations, not through HDR. Finally, CRISPR-Cas-Rx functions differently from CRISPR-Cas9 because it edits RNA instead of DNA, making it have a more temporary effect. Researchers tested the effects of CRISPR-Cas-Rx in human cells, mice, and pigs. Results revealed 50% lower mutated protein levels in cells, as well as reduced mutant proteins and motor improvements in mice. Similarly, pigs also revealed motor improvements and reduced neuroinflammation. The three methods have their unique benefits and challenges, with NHEJ being efficient but imprecise, HDR being precise but difficult to ensure its application, and CRISPR-Cas9 being safer but less well known and could also have potential off-target effects. While the future research could enhance the safety to progress towards a clinical stage, animal model trials have laid a foundation for future research in this field to prevent Huntington's disease and maybe other neurodegenerative diseases in the future. Thank you.